we're going to see insulin secretion and a little bit of the actions of insulin. Remember that insulin comes from a prohormone. This prohormone has two A chain and two B chain. These are 51. The insulin is going to be 51 amino acids protein, and together with the insulin there is a C peptide that is 31 amino acids long. This prohormone that is going to be broken down into insulin and C peptide is storage in vesicles together, so they are secreted in the same molar quantity. Just before being stored in the vesicles, the insulin is post-translation or suffers post-translation modifications that are in the endoplasmic reticulum and in Golgi apparatus, and the next is stored in vesicles. Also, when the insulin is released, also there is release of amylin. Remember that amylin promotes decreasing in gastric emptying, decreasing in appetite, and decreases glucagon. So the insulin secretion is as follows. We have, when there is increase in the ATP to ADP ratio or NAD to NADH to NAD ratio, this closes these potassium channels, and so the membrane potential increases, and there is opening of these voltage gated calcium channels. Calcium goes in and promotes that the vesicles of insulin, vesicles that contain insulin, fuses with the membrane. Also, glucose promotes the production of cyclic AMP, and this cyclic AMP, together with the calcium that is coming here, promotes diffusion. Now the receptor of insulin is, this is a cell. There is one receptor of insulin, another receptor of insulin. Each receptor has two subunits, the alpha and beta. Beta is intracellular, alpha is extracellular. Then there is the hormone comes here, binds to each one of these ones. Then the beta subunits phosphorylate each other. They have tyrosine kinase activity. And next, they come a protein that is called insulin substrate receptor, the receptor of the insulin, the substrate of this insulin receptor, and this tyrosine kinase phosphorylates the tyrosinase residues that are here. And next, there are some other proteins that come here, CH2, and activate many pathways. In the insulin pathway, we have mitogen pathway, that at the end there is activation of the mitogen activated protein, and we have the pathway of phosphokinase 3. This phosphatidylcholine 3 kinase promotes the fusion of vesicles that contains the GLUT4, and so there is expression of the GLUT4 in the membrane of adipocytes tissue and muscular cells, and also promotes the dephosphorylation, the metabolic activities of insulin. Remember that insulin is an anabolic hormone, and the mitogen activated protein or the mitogen pathway goes into the growth effect of insulin, like transcription factors, etc. And in this case, there is a little bit of phosphorylation. But for metabolism, you think as insulin as being a hormone that phosphorylates or promotes the phosphorylation. The insulin acts mainly in the adipose tissue and muscular cells in order to, well, not in order, but it promotes the expression of GLUT4, and it also acts in the liver due to the action of glucokinase and phosphofructokinase 2 that is activated by insulin. Insulin release is promoted first by glucose, next by glucose insulin depending. First is one is glucagon-like peptide, and the other is glucose-dependent insulinotropic peptide or gastric inhibitory peptide. Medications are sulfonylureas, beta-adrenergic stimulation, and decreased alpha-adrenergic stimulation. This beta-adrenergic stimulation, remember that these beta-adrenergic receptors are G-coupled, and so there will be increase in cyclic AMP that releases the insulin. Next, things that decrease, and also vagal stimulation. Vagal stimulation, hormones, secretin, and gastrin. And things that inhibit the insulin release, some drugs would be phenytoin and vinblastine, and the alpha adrenergic response. Insulin promotes glucagon decreases, but glucagon releases sometimes insulin. We will talk about this in other videos. And in the next video, we're going to talk about glucagon.